Hello, my name is Blake Smerigan. I hope you are all doing well. I'm going to be presenting the findings for a two-phase anaerobic co-digestion of food waste and electrocoagulation sludge with a novel startup procedure using activated carbon. This project was conducted by myself, Benjamin Thomas, Sibel Uladog-Demure, and Wei Liao. The goal of this experiment was to find an efficient way to dispose of food waste and wastewater sludge using anaerobic digestion. In order to achieve this goal, we added an initial dose of activated carbon and anaerobic digestion inoculum. We tested to see if this would improve the overall efficiency of the reactors as well as improve the performance of the reactors in their startup periods. We also determined the optimal ratios for the feeding rates of the co-digestion substrate. Finally, we tested to see the influence of different pretreatments of food waste on AD performance. Why did we pre-treat the food waste? Food waste is challenging for AD reactors to handle. It has a low pH that prevents methanogens from populating inside the reactor, preventing methane production. It also proves challenging to break down into the components necessary for biogas production. This is seen 10 to 15 days into the operation. The pH of the food waste fed reactors dropped to very acidic conditions, which corresponded to the decrease in production of methane. The food waste reactors had similar methane production to the control AD effluent fed reactors, which had about 5 times less organic loading. Seen here is the process flow diagram for our experiment. Food waste was fed into the phase 1 reactor at 10% total solids. The phase 1 effluent was then mixed with electrocoagulation sludge that was collected from the purification of concentrated wastewater called black water. This was done in a 1-to-1 ratio by volume and fed into the phase 2 reactors. The final products measured were the biogas produced in the phase 2 effluent. Phase 1 was the acidification of the food waste operated at thermophilic conditions of 50 degrees Celsius. It was semi-continuously fed food waste that was prepared according to a paper describing typical household food waste. The concentrated food waste can be seen on the right. The reactors were constantly mixed at 150 RPM with an HRT of 10 days. Phase 1 increased the volatile fatty acids present in the effluent. It also produced biogas, but the volume of gas produced was not measured. The total solids remained consistent, although the volatile solids decreased slightly in the effluent. The soluble chemical oxygen demand also decreased in the effluent. These values may have decreased due to the production of biogas in these reactors. The total nitrogen was found to increase in the effluent, while the total phosphorus, ammonia, and pH all decreased. Phase 2 mixed the effluent from phase 1 with the blackwater sludge seen on the left. The setup seen on the right shows how water displacement was used to measure the biogas production for this phase. Phase 2 operated at an HRT of 20 days, 15 days, and 10 days. It ran with continuous mixing of 150 RPM at 35 degrees Celsius. The reactors were fed semi-continuously in a nitrogen-filled glove box to prevent exposure to oxygen. The reactors of note for this experiment were fed with pre-treated food waste only, pre-treated food waste and EC sludge, and acidified food waste with EC sludge and activated carbon at 2 or 4% concentration. These conditions for both phases were not what we initially started with. The reasons for these changes will not be discussed in this presentation. If you have any questions regarding these modifications, please ask and I'll explain why these decisions were made. Seen here is the biogas production over the course of the experiment. Some important things to note are the similarities between the 2% and 4% activated carbon with food waste and EC sludge reactors. There was a feeding error in the 10-day HRT that led to the difference between these reactors that will be discussed further. Also, there was a noticeable change in biogas production rate that can be seen by the change in slope between the 20-day and 15-day HRTs. The control AD inoculum, activated carbon, and food waste reactors were terminated shortly after the experiment start due to poor performance and reactor failure. The control food waste with EC sludge reactors succeeded but produced significantly less biogas than the activated carbon added reactors. Shown here is the methane composition for the food waste and EC sludge reactors with 2%, 4%, and without activated carbon. For the 20 and 15-day HRTs, the 2% and 4% reactors had very similar methane composition that was larger than the carbonless control. There is a large difference between the 2% and 4% reactors for the 10-day HRT that is believed to be caused by a feeding of only phase 1 effluent to the 4% reactors. This greatly dropped the pH of the reactors and caused them to fail. This slide shows the hydrogen sulfide concentration in the biogas, which is an unwanted product of anaerobic digestion. There is normally a higher concentration of hydrogen sulfide in food waste fed reactors. Both the food waste with EC sludge and activated carbon reactors had no hydrogen sulfide production until the 10-day HRT. The food waste and EC sludge only reactors had hydrogen sulfide present in their biogas. The pH of the substrates in reactors remained fairly consistent throughout all HRT. In all HRT, the phase 2 effluent of food waste and EC sludge and 2% activated carbon had a higher pH than the previous substrates and effluents. The food waste with EC sludge and 4% AC results are not plotted as they were very similar to the 2% results. The total solids of the food waste with EC sludge and activated carbon was about 20 grams per liter for all HRT. This was a significant decrease from the phase 2 substrate, which shows the ability of the reactors to process the available organics. There was also a large decrease in the volatile solids in the process. The decrease shows that the reactors were highly efficient in the biogas production with the available organics in the system. There was also a large decrease in the soluble chemical oxygen demand in the phase 2 effluent. All of these parameters show that the reactors were healthy and highly efficient in their biogas production. Shown in this table is the change in composition of the measured parameters inside the food waste and EC sludge with 2% activated carbon reactors for a 20, 15, and 10-day HRT in comparison to the phase 2 substrate. As seen on the graphs, there was a large decrease in total solids, volatile solids, and chemical oxygen demand. The total nitrogen and phosphorus decreased, except for the phosphorus in the 15-day HRT. The amount of ammonia was found to increase in the 15 and 10-day HRTs, but was not measured for the 20-day HRT. The pH was about neutral for all HRTs. When comparing the HRT results, the largest biogas production rate was for the 15-day HRT at 730 milliliters per day at a reactor volume of 400 milliliters. This corresponds to a production rate of 1,825 milliliters per day for each liter reacting. For an HRT of 20 days, only 1,235 milliliters per day for each liter reacting was produced. The HRT of 10 days had a similar biogas production of 1,820 milliliters per day for each liter reacting. However, it had a lower biogas production rate of only 420 milliliters of biogas per gram of volatile solids provided. This is lower than the 582 milliliters of production for a 20-day HRT and 644 milliliters of production for a 15-day HRT. The HRT of 15 days also had the largest methane production per gram of volatile solids, where the 10-day HRT had the lowest production per gram. This shows that although the 10-day HRT produced similar amounts of biogas to the 15-day HRT, it was less efficient in utilizing the available organics. From this experiment, we were able to conclude that anaerobic inoculum with activated carbon helps the startup performance of the digesters. Also, the efficiencies of the digesters were improved by the initial dose of anaerobic inoculum with activated carbon. We found that an acidogenesis phase was optimal for the preparation of the food waste. The best HRT for the second phase was a 15-day HRT. We found that there was not a significant difference between adding 2 or 4% activated carbon. Further volatile fatty acid and DNA analysis will be performed in order to get a better understanding of the reactions occurring at each stage and the bacteria and archaea present in the reactors. We used many references to help the design of the experiment, but there were 3 main sources that helped in our decisions. We used reference 1 to design our food waste composition. References 2 and 3 helped us to choose the design conditions of our reactors. I would like to thank Dr. Liu and James Dusenberry, as this research would not have been possible without their help. I would also like to thank the Department of Defense for funding this research. Additionally, I cannot express my gratitude to all of my fellow lab members, as your contributions made this project manageable and it couldn't have been done without you all.